Very good day, children. Today we are going to see the 9th lesson, Applications of Biotechnology. In this lesson, we are going to see the topics, the first 3 topics, that is the applications in medicine, gene therapy and stem cell therapy. So, let us go deep into this. Biotechnology. So, first of all, in this, we have to have a knowledge, we should get enriched with the knowledge of biotechnology, protein synthesis, structure of DNA, RNA and genetic engineering. It is a very must, I should say that it is a must that you must have a knowledge of biotechnology, protein synthesis and genetic engineering as far as this lesson is concerned, because all these are the basics. Unless and until you have a knowledge about biotechnology and genetic engineering. What is genetic engineering? That is, which is also called as the modification or genetic manipulation, that is the direct manipulation of an organism's genes using biotechnology. Then you should know what is biotechnology. Then it is a technology that covers various techniques for using the properties of living things to make the products or provide services. So, that is the agenda of biotechnology. Another one, apart from that, you have to learn even about the structure of DNA and RNA also. The structure of DNA and RNA, what is the purpose of DNA, how many chains are there in that, and RNA. All these you must have a basic or fundamental idea about that. Then what is the use of the protein synthesis? Why should we go for protein synthesis? That is, the protein synthesis, it is a process in which the polypeptide chains are formed. Polypeptide chains are formed from the coded combinations of a single amino acids inside the cells. So, all these knowledge are essential to learn about this lesson. Not only that, you must have knowledge about the recombinant DNA technology also. So, here you will be seeing this, the Escherichia coli. There you have the extra chromosomal substance, what do you call it as the plasmid, which is said to be the boon for the biotechnology for the human beings, especially to get recombinant DNA technology products given by that. So, all the, not only that, also the chromosome structure, everything can be modified by joining together of a DNA molecule from 2 different species. And this one, the DNA molecule gets inserted into the host organism, and then you can produce new genetic combinations thereafter. And you can use this particular genetic new combinations for the value of science, medicine, agriculture and industry. So, the basic steps you should learn, you should have an awareness about that, and apart from that, you should have a knowledge about the animal biotechnology and plant biotechnology also. That is animal biotechnology. What is animal biotechnology? That is a set of techniques by which the living creatures are modified for the benefit of human beings, that is for the other animals also. Not only that, the agricultural development, plant biotechnology is nothing but the agricultural development. Mainly it is for the, everything is only for the humans, the modifying plants and animals, everything with the desirable traits. So, we have the animal biotechnology, plant biotechnology. So, all these are the fundamentals you should learn, you should have a knowledge about this. Only then you can enter into this lesson to study about the rest of the topics. So, the first topic what we are going to see here is applications in the medicine. So, here medicine is the first one, the recombinant DNA technology you can see here, which has led to the large scale production of various hormones and proteins of therapeutic use. What do you mean by therapeutic use? For the medicinal purpose, to cure the diseases, whatever it is. For that you need to have various hormones and proteins. That is why we go for this applications in medicines using DNA or DNA or recombinant DNA technology, anything you can say. Next one is the production of recombinant human insulin, recombinant human insulin. So here actually the human insulin is synthesized in the beta cells of islets of Langerhans in the pancreas. So here you can see the structure of pancreas in the human being. Here near the duodenum, at the C-shaped structure of the duodenum, you can see the structure of pancreas here, and the enlarged structure of this islets of Langerhans. Inside you can see the beta cells, alpha cells and beta cells, all these cells are present. So in this we can see the beta cells alone, they are secreting the amino acids, so which is formed of 51 amino acids. The insulin, which is formed of 51 amino acids arranged into 2 polypeptide chains of A and B. A and B you can see here. This is the polypeptide chain of insulin. Next one, the polypeptide chain of A. What about the A? The structure of A, here you can see it is formed of 21 amino acids. So here this A is formed of 21 amino acids, and the polypeptide chain of B is formed of 30 amino acids. So both are held together by disulfide bonds. Disulfide means 2 sulfide bonds. Okay, so what is the purpose of this insulin? Actually, why do we go for insulin? Why do we take interest in insulin is that it controls the level of glucose in the blood and also facilitates the cellular uptake and utilization for the glucose, utilization of the glucose for the release of energy. So energy comes from the glucose. For that you have to keep up the insulin in the normal level in our body. Okay, next after that, if there is deficiency, what will happen? If there is deficiency of insulin, this might lead to a disease, the diabetes mellitus, very characterized by the increased blood glucose concentration, and a complex system which may lead even to the death if it is not treated properly. Okay, so for this you have to go for insulin dependence program, continuously to treat this deficiency. So that is the main agenda. Formerly, insulin has been isolated and purified from the pancreas of pigs and cows. So here you can see the picture. This is the structure, the anatomy, inside structure. You can see in the stomach you can see the pancreas. So here you can see the pancreas. This is a collection from pigs. So here you can see the cells which is identified, and actually the insulin was previously isolated from the pigs and cows, that is the bovine, to treat the diabetic patients. So there is a minor difference. Actually we have to say a minus difference, isn't it? The structure of animal insulin compared to the human insulin. So which resulted in the allergic reactions of some of the diabetic patients. We will see why there is a difference. What makes the allergic action of the bovine and human insulin? What is the difference between that? Why there is an allergic action resulted? So here you can see the A chain and B chain of the bovine. The reason is that there is a difference. So the human insulin, the first one in this, the threonine and alanine, there is a change in the position in A chain position 8 and A chain position 10. You can see the isoleucine in the human insulin and valine in the bovine insulin. See, there is an alternative replacement of the A chain and B chain in the 8th position and in the 10th position. Then the B chain in the 30th position comprises threonine in human and alanine in bovine. So like this, there are alternative changes which was found later, and so this was, they identified that this was the reason for getting the allergic action. Then what happened after that? Now the production. Now they have entered into the recombinant DNA technology, and they were able to produce the insulin with the help of rDNA technology by the insertion of human insulin gene on the plasmids of E. coli, that is the Escherichia coli. See here the DNA. In the DNA you have the pancreas cells, human pancreas cells, you can see the DNA here, and also the bacterium Escherichia coli, you have the extra chromosomal substance, what do you call it as the plasmid DNA. So in this, by splicing technique, they were able to get back the position of this particular gene, and they were able to introduce this into the recombinant gene. Now they were able to clone all this and it is multiplied, then we are able to get a human insulin. So now this production of insulin by recombinant DNA technology involved the insertion of human insulin on the plasmids of the bacterium, what do you call it as? E. coli. Now it is manipulated, we are able to get the human insulin. What about the steps? The polypeptide chains are synthesized. So how are they synthesized? See, here they are all synthesized by precursor cell. So first of all, to synthesize this, we need a precursor cell. What do you call it as? Pre-pro-insulin. This pre-pro-insulin, which contains two segments, that is A segment and a B segment. See here the color, the blue one is the A and the orange one or yellow, you can see what you see here is the B chain. Now to link this you need a C chain. Okay, now you have already a chain and a B chain is there. To link these two we have a C chain. Now this should be guided by a leader sequence. So you have the pink color is the leader sequence. So to translate the genetic material, for that the pre-pro-insulin is used, and now after the translation is over we are able to get the pro-insulin structure. Then finally after the completion of the translation, the C chain is excised. It is excised, leaving the A and B polypeptide chains. So now we have the insulin. Now it is ready. This insulin is now in this was the first ever pharmaceutical product, we could say, pharmaceutical product of recombinant DNA technology administered in the humans. Now the approval, what about the approval? See, whenever you get a medicine, you have to give a trademark, you must get the patent for that. So that patent is given, approval is given to use this recombinant insulin for the diabetes patient that is suffered by diabetes mellitus. That was given in 1982, and only in 1986 the insulin was marketed under the trade name Humulin. You can see this, Humulin. So this was the one what we are using now. Okay, the second one is the interferons. What are interferons? The question is, what are interferons? They are nothing but the proteinaceous antiviral species specific substances produced by the mammalian cells when infected with the virus. So when are they produced? Only when they are infected by the virus, they were able to produce the interferons, which are nothing but the proteinaceous antiviral species specific. So that point you have to keep in mind, this is species specific. So this was first discovered by Alec Isaacs and Jean Lindman. You can see the picture here. So based on the structure, they classified interferons as alpha, beta and gamma interferons. You can see the picture here, interferon alpha, beta and gamma. So what is the purpose? They stimulate the cellular DNA. So the DNA which are present at the cellular level, they are stimulated to produce the antiviral enzyme. That is the agenda. To produce antiviral enzymes which inhibit the viral replication. We have to stop the viral replication. So that is the agenda. To stop the viral application, what we have to do is that you have to apply this particular interferon. So here you can see that the enzymes which inhibit viral replication and protect the cells. So our purpose is fulfilled by the usage of this interferons. Another one, there is a similar factor, that is the factor 8, which we use for hemophilia to describe that disease. There is missing character in the hemophilic persons, that is the factor 8. The interferons could be isolated from the blood, similar to the hemophilic patients. What do we do for diagnosis? So here also we can isolate the interferons from the blood, but here you need the amount of blood required for the isolation is enormous. We could say it is enormous. So such an enormous amount of blood is not practical, isn't it? So we cannot afford. So this is not considered properly. And here, to overcome this issue, what do we have to do? what do we have to do? Then you have to go for an alternative, isn't it? So now we are applying this rDNA technology using the other alternative as a Saccharomyces cerevisiae. Saccharomyces cerevisiae, that is the yeast, which is a suitable production of recombinant interferons. We were able to produce recombinant interferons with the help of Saccharomyces cerevisiae, that is the yeast, other than the E. coli. Escherichia coli, we are not going for that. We don't want E. coli anymore. We have an alternative, what do you call it, as the Saccharomyces cerevisiae, the yeast, which is serving this purpose. But why we are going for the yeast, that is Saccharomyces cerevisiae? Because there is no machinery for glycolization. In E. coli, you don't find the machinery for glycolization of proteins. So that is not seen in E. coli, whereas it is very easier in the case of Saccharomyces. So you can see the picture here, how they were able to get in each and every steps here. From this, the first one, it is entered inside. After that, the step by step, the production, glycolization process. Then finally, how does it enters? So our agenda is to get the interferons, by rDNA technology. Okay, so what are the diseases caused due to the suppose if interferons is not properly present, if interferons is not there, what are all the diseases we can treat with the help of interferons? So the first one is a cancer, AIDS, herpes zoster, genital warts, multiple sclerosis, hepatitis. So all these can be cured or it can be treated with the help of interferons in human beings. Another one, in spite of the therapeutic applications, so this is not with a easily approachable medicine, so that anybody can get. Even the normal man cannot get this particular facility. So for a common man, this is of a very high cost production, so not easy for them to get. Okay, so next we go for this recombinant vaccines. So here the technology has been used to produce new generation vaccines. Day by day our technology is improving, genetic engineering, we have so many aspects. So in that we are producing new generation vaccines with the help of rDNA technology. So here the limitations. So earlier we have the traditional vaccines where there are limitations for the productions. But here it is not so. So you can produce the vaccines generally for uniform quality and with the less side effects. That is what we expect, isn't it? Less side effects. And when compared to the vaccines produced by the conventional methods. Earlier we have produced so many vaccines by conventional methods. So here the uniform quality we need and also the less side effects we expect, isn't it? So here are the types of vaccines produced by the viruses. You can see that live attenuated virus-like particles. So like this you can zoom here and see what are all the products which are produced by this. And three types of recombinant vaccines are seen here. One is subunit vaccine, another one attenuated vaccines, the third one DNA vaccines. So these are the main three types. So in this, the first one which we are going to see now is the subunit vaccines. That means they are the components of pathogenic organisms. Actually the vaccines that use the components of the pathogenic organism. What is a pathogenic organism? First of all, you should learn about that. The disease producing organisms are called as pathogens. So those organisms are called as pathogenic organisms. So here we use the whole organisms which are called as the subunit vaccines. So here the vaccines which are produced from the pathogenic organisms rather than the whole organisms, we call it as the subunit vaccines. So here this is very much suited for developing a new subunit vaccines. So it's very much suited for the new subunit vaccines which include the components. What are the components we use? Proteins, peptides, DNAs. All these are used from the pathogenic organisms to create this subunit vaccine. Now, what are the advantages? We should have some advantages when we introduce a new product, isn't it? That is what the persons expect. Here, the purity in the preparation. First of all, you can find the purity in the preparation, stability and safe use. So these were the advantages of subunit vaccine. Next one is the attenuated vaccine, attenuated recombinant vaccine. So here this includes the genetically modified pathogenic organisms, genetically modified. So whether it may be a bacteria or a virus, so this is made non-pathogenic. This virus of bacteria are made non-pathogenic, and here they are used as a live vaccine, which were referred as the attenuated recombinant vaccines. Okay, so that is the difference between the subunit and recombinant attenuated vaccines. So here recombinated vaccines. So here one type we have. See here the genetic material and the normal one here. It is spliced, multiplied, then the DNA vaccine is produced. Now that is introduced for vaccination. We have examples as poliomyelitis vaccine and this Engerix B. So these are the vaccines which were used for this recombinant vaccines here. The third one, finally, we have the DNA vaccines. DNA vaccines actually genetic immunization. So we go for the process genetic immunization. Immunization means making you healthy. Making you healthy. Whatever the hazardous that is present in the nature, we don't find any other opportunities to get rid of that disease. For that purpose, we go for immunization. So here DNA vaccines, this is a genetic immunization by using DNA vaccine. Is a novel approach which we are finding now in 1990s. It has started. Now the immune response of the body is stimulated. Now this is stimulated by the DNA molecule. And after that, this is, you see here, this is relatively easy and expensive design and produce. Very easy for one, and is expensive also. At the same time, these vaccines, they were produced by the new techniques for definite advantages. We have a definite advantages. You can assure the advantages by this, by producing the target proteins, because they aim for the target proteins, long lasting immunity and trigger immune response. So soon after that, the immune response is triggered, and so they are only against specific pathogens. You see here, they are against specific pathogens with less toxic effects. That is what we expect when we go for vaccination, isn't it? So that is fulfilled by DNA vaccines here. See here, how does it, how do the process takes place here? DNA vaccine plasmid is here. So you can see the eukaryotic plasmid enhancer is there, gene coding pathogenic protein, both are combined. So it is able to produce a new level. Like DNA vaccine, this consists of a gene coding and antigenic protein which is inserted onto a plasmid. Gene coding and antigenic protein. Okay, now this is inserted into a plasmid, and then this is incorporated into the cells and then into a target animal. Whichever the animal you need, the target animal, this is inserted. Okay, after that the DNA, now this is instructing, this is giving instruction to the cells to make antigenic molecules. So after that, after the manipulation, it is giving instruction to the molecules which are displayed on the surface. So here this will be displayed on the surface. See, antibiotic resistance gene, everything will be displayed on the surface. Okay, so after that what will happen? Now this would evoke the antibody response to the free floating antigens. See, sometimes they think there is no antibody response for them, they will be freely floating over there. Now this will evoke the antibody response, thereby what will happen? The cells will start secreting the response, immune response, and now the DNA vaccine, you can see here, they cannot cause the disease. DNA vaccine, they cannot cause the disease here. They contain only the copies of few of its genes, because that's why it cannot cause a disease. Okay, so here everything is produced. Now this is ready for use. Now the DNA vaccines, we expect it is easy and inexpensive. Now it is easy and inexpensive to design and produce. Okay, so another example for that subunit vaccine, we have this recombinant hepatitis B vaccine, which is an example for a subunit vaccine. It is done by cloning the hepatitis B surface antigen. So hepatitis B surface antigen is cloned here. So after cloning, we were able to get multiplications of this. Then it is sent to fermentation tank, and then it is ready for the use, the HP vaccine here. So here also we are using Saccharomyces cerevisiae, the yeast, as an alternative for E. coli. Okay, this is another example. So next topic which you are going to see here is the gene therapy. So in gene therapy, first of all, you can see there are some diseases which cannot be cured, There is no alternative. Only death is the final destination, they say. But now this is not so the case. You can find the gene therapy as an alternative treatment. So here, for hereditary disease, I could say it is for the hereditary disease, this can be cured with the help of gene therapy. Okay, the transfer of a normal gene, you are transferring a normal gene, see the picture here, a healthy gene, a normal gene to the person who carries one or more mutant alleles. So if a person is having a mutant allele, for them you can transfer the normal genes. Okay, so this expression of the normal gene in the person results in the functional gene product. So now you have the functional gene product here whose action produces a normal phenotype. Okay, so this is what we expect, isn't it? As far as hereditary problem is concerned. So delivery of the normal gene is accomplished by using a vector. For that we use a vector. We need a vector. What is a vector? Which is carrying the molecule which is inserted inside for therapy. So here the vector which may be Saccharomyces cerevisiae plasmid or E. coli plasmid or Saccharomyces cerevisiae the yeast. Okay, so this is used as a vector to cure certain diseases like the cystic fibrosis. The main gene therapy here for correcting the single gene mutations, what do you call it as the cystic fibrosis and hemophilia. Cystic fibrosis, you can see the genetic systems. See some of the genetic disease that causes persistent lung infection. This is concerned with the lung infection. So and the limits of the ability to breathe over. So they get suffocation over time because in people with the CF mutations in the cystic fibrosis, the transmembrane, you can see the transmembrane conductance regulator gene, that is the CFTR gene, they cause limitations for breathing. So that membrane is not having the property to breathe easily. Okay, so fibrosis is caused due to that. For that you can make the correction by replacing the gene there. Another one, hemophilia. Hemophilia, what do you call it as hemophilia? See, for a healthy person, see when you go for sharpener or when you sharpen with the pencil, with a blade, you get cut off, a small cutting. Okay, after that you get bleeding. That bleeding will be stopped within a fraction of seconds. For everybody this is a normal condition. But in the hemophilic persons, this is not so the case. See here, for the normal person's condition and for the hemophilic person's condition. So here, this is, they were unable to produce, a delayed fibrin clot is given. So immediately here, the plug is formed here, okay, in the normal persons, whereas here, the clotting factor, a very weak platelet is present. So that plug formation is very much delayed. They were able to get only the incomplete fibrin clot. That is why the bleeding is continued. So you call the hemophilic persons as the bleeder's disease. So they are, that is mainly due to the correction which has to be done with the 8 factor. Okay, the factor 8 is responsible for that. Actually we call it a zigzag inheritance, which is passed from male to male through female, isn't it? So here you can see the thrombin, the factor which is necessary for clotting, which is delayed here. So two types of hemophilic, that is A and B said, whereas A is said to be very, very dangerous. Okay, so that can be cured with the help of gene therapy. That's what I'm coming to say. Okay, there are two strategies we have. Two strategies. One is gene augmentation. You have to multiply it. Okay. Another one is gene inhibition. That is gene augmentation, when I say, involves insertion of a DNA into the genome. Insertion, you are inserting it. Insertion of a DNA into the genome to replace the missing gene product. Whichever the missing gene product is there, it can be inserted. Okay. So another one is inhibition. In the inhibition, insertion of antisense. Antisense which inhibits the expression of a dominant gene. So that antisense will inhibit the expression of a dominant gene. So that you call it as inhibition therapy. So these two are the strategies of gene therapy. One is gene augmentation, another one is gene inhibition therapy. So, two differences are there between the somatic cell gene therapy and germline gene therapy. Somatic cell gene therapy, you can see the gene transferred into the somatic cell, as the name implies, somatic cell. Okay. Another one, gene germline means it is the reproductive cells or the germ cells. This refers to the germ cells, therapeutic genes. So, here gene transfer is done in the somatic cell, gene transfer is done in the germ cell. Another one, where can you introduce this? This can be introduced into the bone marrow cell and the blood cell, skin cell, whereas here germ cells you have to introduce only in the egg or in the sperms. Okay, they are the genes. In those genes alone you can insert. Next one, this will not be inherited into the later generation because this is only a somatic cell, whereas here this is heritable because passed on to the next generation. So, these two differences you should learn. The next one is the process of gene therapy with reference to the SID, that is the severe combined immunodeficiency. See here, the problem is with the ADA gene, that means the adenosine deaminase gene, which is coding is not functioning here. Not functioning in the sense it is not producing the normal T lymphocytes. So, these lymphocytes cannot elicitate the immune response in the children. So, what is the right approach is that to give the functional ADA to the patients. Okay. So, this can be cured by the bone marrow transplantation and the defective immune cells are replaced with the help of a healthy immune cells from a donor. So, this is done mainly by the enzyme replacement therapy. Okay, that is what we go for here, enzyme replacement therapy. So, you can see the structure from the bacterium, plasmid, a part is removed and there the insertion is done for ADA gene and then it is introduced into the retrovirus, their cloning is done. After that, the lymphocytes, it is inserted into the normal lymphocytes, then it is manipulated and that is given to the genetically altered cells or re-implanted to produce ADA in the person who are affected with the SID. So, normally these persons are said to be having a very short lifespan. If it is introduced into the young stage, they do not have to go for further treatment because this will be proliferating in the blood, whereas in the case of adults, every time they have to go for the gene therapy. Okay. So, this is one example due to SID, the adenosine deaminase enzyme production is not functional. The next one is stem cell therapy. Stem cell therapy is nothing but the stem cells. What are stem cells? Here they are nothing but undifferentiated cells. They are nothing but the undifferentiated cells which are found in the most of the multicellular animals and maintain the undifferentiated state. We want only the undifferentiated state of a cell even after undergoing numerous mitotic divisions. Even after undergoing mitotic divisions, never mind, still it remains undifferentiated. That state only we need for the stem cell therapy. So, here the stem cells, they have the potential for the ability to regenerate the damaged and diseased organs. So, that potential only we need from the stem cells. So, here you can see the pictures were given here. So, you can zoom and see the structures which are all formed directly by the stem cell therapy. What are all the characteristic features of the stem cells is that the undifferentiated cells, they will produce whatever the cells which you need, target organs, that is the nerve cell, blood cell, liver cell, muscle cell, whichever is necessary to replace or the damaged tissues, capability of self-renewal, exhibit cellular potency, efficient to differentiate into other cells. So, these are the characteristic features. So, they are capable of self-renewal and exhibit cellular potency. So, that is the main agenda we have to see. What are stem cells? What is the capacity? They are capable of self-renewal and exhibit cellular potency. And next one, stem cells, they differentiate into all types of cells from the 3 germ layers. What are the 3 germ layers? We have ectoderm, endoderm and mesoderm. So, they have the capacity for that. And they can differentiate into the main organs, target organs. So, another one, mammals, 2 main types of cells are present here in mammals. What do you see here as the ES cells, what do you call it as the ES cells or embryonic stem cells, the ES cells and the adult stem cells. ES cells are said to be pluripotent. That is, they can produce 3' germ layers, ectoderm, mesoderm and endoderm. You can see here, one picture is given here in vivo. The pluripotent cells, they were able to differentiate into the respective cells, whichever you need. And another one, embryonic stem cells are said to be multipotent. Pluripotent is differentiated from the multipotent cell. Here, multipotent stem cells that can differentiate into number of types of cells. So here ES cells, they are isolated from the epiplast tissue. Where do you find epiplast? In the blastocyst, isn't it? In the blastocyst you can find the epiblast tissue, the inner cell mass of the blastocyst. From there it is isolated. So we stimulate, and by this 200 types of cells were produced from the adult body. So ES cells, they are said to be immortal. You call it as immortal because they can proliferate in a sterile culture medium. They don't need any other sources. They can get proliferated or multiplied in the sterile culture medium, maintain their undifferentiated state. That is what we expect from the stem cells. So what are the sources of stem cells? We can get it from placenta, umbilical cord, amniotic sac, amniotic fluid, bone marrow, blood and epiblast of embryo. So these are all said to be the sources of stem cells. The next one, the adult stem cells. Where do you find here? If it is found in various tissues of children and as well as in the adult, it may be from the somatic stem cell, which can divide and create another cell similar to it. Okay. So the multipotency mainly for repair system of the body, replenishing adult tissues and for all this. And the red bone marrow, we can call the red bone marrow, which is a rich source of adult stem cells, and the potential application, that's what we expect from the human stem cells for the generation of cells and tissues for cell-based therapies, mainly for cell-based therapies. Human stem cells could be used to test new drugs also. For new drugs also we use human stem cells. So the stem cells, you can see the blastocyst here, also the cells from the adult stem cells, embryonic stem cell. You can see the differentiation. Embryonic stem cells are pluripotent and are capable of to differentiate into any germ layers, whereas adult stem cells are multipotent and they are capable of differentiate into any of the related cells. Okay, this is the difference between these two. Next you have the cell banks, the stem cell banks. You might have heard about some of the ad recently in TV. They would have shown some ad that you have to store your stem cells. Please donate stem cells. Please donate the cord cells. All this they used to advertise in TV for stem cell banking. What is the use of this stem cell banking is that the extraction, processing and storage of stem cells. What is the use? Which is mainly for the treatment of in future, if the same person who have donated the stem cell is having any problem, so that time this stem cells can be used. For that we store these stem cells in the bank. What do you call it as stem cell banking. So when required it is used. So here amniotic cell bank is one type. We can find the amniotic cell bank. So this amniotic cell bank stores the stem cells derived from the amniotic fluid for the future use. You can see here by amniocentesis method, this is taken out from the amnion, and these are specifically used for the individual, and for banking cost you have to pay for that. For storage you have to pay for the banking cost, and also from the card banking, blood banking is also done there, stem cell. So here it is extracted, processed and then storage. Storage in cryopreservation is stored for future use. Cord blood banking is also done. What do you see here, the cord when it is cut after the parturition. So that is mainly used in having so many stem cells, undifferentiated cells, which can be used for future use. So here the extraction of stem cells from the umbilical cord during the childbirth is taken, which is more valuable than anything. So here the most popular source of stem cells, we could say the umbilical cord or the cord blood. So this can be got from placenta, amniotic sac and amniotic fluid. Everything is said to be rich sources in terms of both quantity as well as quality. Okay, so far we have completed gene therapy. We'll see you in the next class. Thank you.